Chalo, in my last class, we learned about the physiology of menstruation and ovulation. What comes next? Of course, the fertilization. So, we will be discussing fertilization in this different headings. We will define fertilization and its physiological significance, gamete transport and its capacitation, and we will talk about the acrosomal reaction and Zona Pellucida binding, and we will describe about the cortical reaction and prevention of polyspermy. What is polyspermy? Any idea? Batao. Anybody having any idea about polyspermy? One egg will be fertilized by one sperm. Once one sperm is fertilized, no other sperm can enter that particular egg. So there will be no multiple sperms. So polyspermy, we will be discussing about that. And we will also explain the formation of zygote and early embryogenesis and clinical correlations. To start with the overview of fertilization, it is the fusion of male as well as female gametes to form a diploid zygote. What is diploid? What is haploid and what is diploid? We will come to that. It always occurs at the ampullary portion of the fallopian tube, which we have already discussed in the previous classes. Whenever the egg is released from the ovary, that fallopian tube, it sucks inside that. It sucks the ovum inside the tube with the help of the femoral and of the tube. Okay? And the first, after the femora, what is that part? That swollen part, that is the ampullary portion. So, all the fertilization usually takes place in the ampullary portion of the fallopian tube. And it requires, what does it require? It requires a synchronized maturation of both male as well as female gamete. Okay? So, how the sperm is being transported? In every ejaculation, it releases around 200 to 300 million of sperms. Okay? And the capacitation occurs in the female genital tract. They are immortal. When the semen is deposited in the posterior fornix of vagina, those sperms are all sleeping sperms. Okay? Gradually they enter into capacitation only when it is inside the female genital tract. That means it is getting activated. Once it is released from the male, it is not activated that moment. Okay? Only when it is deposited in the female genital tract, they get activated, which we call as capacitation. And gradually they attend to hyperactivation. They become hyperactive very fast. Okay? And aids penetration through the cumulus ophorus. Where does this cumulus ophorus is? It is the outer cell, one cell line that covers the or guards the egg cell. That is called cumulus ophorus. Only around 100 to 300 of sperm reaches the ampullary portion of the tube. Even though around 200 to 300 millions are deposited inside the vagina, however, only around 100 to 300 will reach the ampullary portion of the tube where the fertilization will take place. Okay? And how does they reach? The capacitation and becoming hyperactive. And they start swimming up from the vagina to the cervix to the uterine cavity and towards the tubes on either side. And they reaches the ampullary portion. How much? Only around 100 to 300. And then how the oocyte is transported? As we have discussed in earlier classes, there is an ovulation and then this femoral end, finger-like projections, we call it femoral end. They sucks this egg from here and then they brings it here. They picks it up, and then the sperm travels through the whole tract and reaches the ampullary portion here, and they meet each other here, and if the sperm fertilizes the egg here, and gradually by 4 to 9 days it keeps on traveling and reaches here in the endometrium, which we call implantation, and we'll talk about it. And it is picked up by the femoria and transported by the ciliary action. What is the ciliary action? Is a small projection that keeps on moving like this, and a peristaltic movement. Both acts together. And the egg cell is surrounded by cumulus cells and zona pellucida. We'll gradually come to that. So, capacitation, we have already spoke about it, the functional maturation of the sperm. Once it is deposited inside the female genital tract, and there is a removal of the glycogen coat. This yellow one is the glycogen coat and the seminal plasma proteins. Once this is released, once the sperm is inside the genital tract, and they gains the motility. And once this layer is released, after depositing inside the vagina, there is an increased calcium permeability that makes it hyperactive. Is it clear? Not clear. This is a normal sperm deposited inside the female genital tract. Okay? This is an activated sperm. Can you feel the difference? The difference is this layer, this purple color and yellow color. It is not here. Is it clear? So once this whole thing is deposited inside the vagina, there is a called capacitation. What does the capacitation means? Capacitation means the release of this outer layer and making it like this. Is it clear? So once this layer is removed inside the vagina, then there is increased calcium permeability, calcium ion permeability. Whenever the calcium ion permeability increases, it reaches the motility. Okay? And next we'll come to the acrosomal reaction. So they have gained the motility inside the female genital tract. Now what they have started? They have started swimming up. From the vagina to the cervix to the uterus and towards the tube, they started swimming up. Okay? The sperm did not get the ovum yet. Once the sperm gets the ovum, meets the ovum, then the acrosomal reaction will come in. And we'll talk about it. Now the sperm was deposited inside the vagina. The capacitation took place there. After removing that glycogen coat, they started moving, and now the sperm has reached the egg cell. Now comes the acrosomal reaction. What is this acrosomal reaction? So, whenever this sperm reaches the egg has got a zona pellucida, outer cell cover that is protecting the cell, the egg cell which is inside. Just imagine a ball, okay, cricket ball. It has got something inside, and outside there is a cover. That outside cover is zona pellucida. Okay. Now the sperm is coming with the head and touching the outer surface of that cricket ball, or you take it as a egg cell. Now once it touches that, it will be binding towards the pellucida glycoprotein ZP3. There are two proteins, ZP3 and ZP1, outside the egg cell, where the zona pellucida is there. Okay? That zona pellucida contains two proteins. One is ZP3, that outer one, and ZP1, that is the inside one. And whenever that ZP3 comes in contact with the sperm head, okay, it binds the sperm head and it releases certain enzymes. What is those enzymes? Those enzymes are hyaluronidase and acrosin. Okay, so now the egg has the zona pellucida has taken this sperm here, because the ZP3 has got the receptors for that sperm. Okay, that the sperm has attached with the ZP3. Where the ZP3, that the zona pellucida. Inside is the egg cell. So once it is attached here, now these enzymes are released What is those enzymes? Hyaluronidase and acrosin, and it breaks the Zona pellucida. Okay? And the sperm enters the egg cell. Is it clear till now? It's very simple. So, ZP3 attaches the sperm on the surface of the egg, and the acrosome acrosome of the sperm releases that hyaluronidase and acrosin, which breaks the Zona pellucida, and the sperm enters the egg, and the fertilization takes place. Okay? That Zona pellucida binding, as I say, it contains two proteins. One is ZP3, that the primary sperm receptor which receives the sperm, and then inside there is a ZP2, that is the secondary binding site after the reaction. What is that reaction? Acrosomal reaction. What was that acrosomal reaction? ZP3 bringing the sperm here, then this is releasing the hyaluronidase and acrosin, breaking the layer, entering inside, and inside who is waiting? Fertilization. So dog sperm can fertilize a dog's egg only. Maybe the breed may be different. Okay? But a human sperm cannot fertilize a dog egg cell. So vice versa. So it's a species specific. Human is human. It may be Negro, it may be white, it may be people like us Asian. But that's called species specific. Any human sperm can get attached with the ZP3 of the Zona pellucida protein, and then can further break it and enter further, reaches the ZP2, and then further reaching the egg cell. So it is always your species specific. So dogs will give birth to dogs, birds will give birth to, mammals, all mammals of that particular specific species can give rise to specific species. Okay? That's why this is called species specific fertilization, because of the ZP3 and ZP2, because the ZP3 is specific for human sperm only. Is it clear? ZP3 and ZP2 is specific for human sperm only. Okay? Maybe blackish, maybe whitish, maybe like us. So that's called species specific fertilization. Is it clear up to this? So the penetration I was already talking about it, the fusion of sperm and the oocyte membrane. What was that membrane? Zona pellucida. Okay? We have broken that, we have entered that inside the oocyte. Now the sperm nucleus, then centriole, mitochondria enters the oocyte cytoplasm. I hope you understand what is cytoplasm. Okay. So that sperm nucleus, centriole, mitochondria, that enters the oocyte cytoplasm finally, breaking the layer of Zona pellucida. Is it clear? I feel it's very simple physiology. If anybody doesn't understand, please raise your hand. Now the oocyte completes its meiosis to thus formation of ovum. Okay, I will come to that. And it releases the second polar body, means sharing out of the chromosomal content. Polar bodies are sharing out, dividing out of the chromosomal content. There are two polar bodies released. One is first polar body, that is second polar body. I will come to that. So is it clear up to this? We have broken down the Zona pellucida, ZP3 acrosomal reaction, then entered further. ZP2 was waiting to receive the sperm, and that sperm entered the egg cell, where it has entered the cytoplasm of the oocyte. Is it clear till that? Any doubt? No. Now comes the cortical reaction. What is that cortical reaction? Once a sperm fertilizes an egg, no other sperm can get into that. How it cannot get into that? because it brings the outer layer very hard. It releases the cortical granules from the oocyte. Granules means dane ka cheese type. Okay? From the oocyte, that particular egg, which makes the enzymatic modification of Zona pellucida. Zona pellucida break karke we went inside, the sperm has gone inside. So that breach is there, that breakage is there. Now that breakage has to be blocked, otherwise second sperm will get inside. Third sperm will get inside. So there will be polyspermy, isn't it? So it has to be blocked. That breakage has to be blocked. We will be blocking that. That is the granules released from the, that's called cortical granules, which are released from the oocyte, and it modifies the Zona pellucida, making the Zona pellucida sealed. One sperm has entered inside. Now the cortical granules are released from the oocyte, and it makes that particular point of entry sealed. Okay? Thus it blocks the polyspermy. Is it clear up to this? Okay. Now coming to the formation of pronuclei. The male pronucleus are formed from the sperm nucleus. Pro means before. Okay? So, male pronucleus forms from the sperm nucleus, and the female pronucleus forms after the meiosis completion. We will come to that. And then comes the migration and fusion, leading to diploid cell. That cell which is capable to go under mitosis and further formation of the embryo. So what is zygote formation? A diploid cell with restored chromosomal number. Diploid cell with restored chromosome number, which may be 46XX in case of female and 46XY in case of male, is known as zygote formation. And when does the cleavage starts? The breaking starts around 24 hours after fertilization. Because the sperm that goes and the egg which comes, they cannot give rise to 46 plus 46, then 46 plus 46 kitna hota hai? 92 plus double X, triple X, Y. It's not possible, na? It, after meeting these two with a fully settled chromosome with either 23X or 23Y, and from the female side 23X oblique 23X, because you don't have an Y. So it has to break down, and once this meets together, then it becomes 23 pairs. If 23 pairs, that means it is 46, and it can be either male or female, that is XX or XY. Okay? And further early embryo, and then starts the embryonic development. Are you getting the sequence? We talked about the sperm deposition in the female genital tract. That reaches the capacitation, starts moving up, went inside, got the egg, and then Zona pellucida was broken by acrosomal reaction ZP3, and then further enters the egg ZP2, and then entered the egg cell. The sperm completely started sharing the genetic material, and everything reaches the cytoplasm of that oocyte. So fertilization has taken place. Now the development of embryo starts. Okay? So there is a cleavage of the cells. You must have learned the mitosis and meiosis, or you are learning now mitosis, meiosis. Is that it? So starts cleavage. 2 cell will become 4 cell. Okay? And the 4 cell will become 8 cell. Okay? And gradually they starts from 2 cells to 4 cells, 4 cells to 8 cells, because it's a mitosis, doubling. Meiosis is half. I had 4, became 2. I have 4 cells, 2 this side, 2 this side. It has broken down to half. So we call it meiosis. Now I have 4 cells. This side also I have made 4, this side also 4. Means I made 8 out of 4. Double. So it's a mitosis. So when 2 cells, a cleavage, it is cleavage to 4 cells. And then 4 cell becomes to 8 cells. And 8 cell become to 16 cells. Whenever it comes to 16 cells, we call it morula. Whenever it is 8 cell stage, we call it blastocyst. Okay? First, it was 2 cells separate, sperm cell, egg cell. They met together, became 1 cell with chromosomal pattern either 46XX or 46XY. Now it started breaking down 46XX to 46XX, if 46Y, XY to 46XY, 1 to 2, 2 to 4, 4 to 8. When it reaches 8, we call it blastocyst. It's because some kind of fluid starts accumulating in between cells. So that's why we call it cyst. Every cyst contains fluid. So we call it blastocyst. And the next stage comes the morula, when it is a 16 cell stage. From 8 blastocyst, 8 cell or blastocyst, when it is again entered the mitosis, so it becomes your 16 cell stage. That's called morula. Okay? And the blastocyst formation is completed by around day 5 to 6 with intracytoplasmic membrane and development of trophoblastic cells. Okay? So, now the embryo was formed. We started from very beginning. Deposition of sperm till fertilization. Now after the fertilization, the embryo has formed. Up to what stage? Blastocyst. Okay? 2 cell, 1 cell to 2 cell, 2 cell to 4 cell, 4 cell to 8 cell. That's called blastocyst. Now it is time for the blastocyst to get implanted. Where it will get implanted? It will get implanted inside the endometrium. Now the blastocyst hatching form the Zona pellucida. So we spoke about the Zona pellucida. Once it has entered, again Zona pellucida was blocked. So that the polyspermy was blocked. Now it is rotating from the femoral end to the endometrial cavity. By how? By ciliary movements as well as peristaltic movement. Now it has reached the endometrial cavity. But the Zona pellucida is still there, isn't it? Zona pellucida is still there. If Zona pellucida is still there, how it can make communication with the mother? That outer shell has to be broken to get implanted and start making communication with the mother. So that's called blastocyst hatching. That removal of that hard cover outside that fertilized egg, that's the embryo, and removing the outside cover, that is the Zona pellucida, and now it will get inside the endometrium. So that's called blastocyst hatching. Okay? Which will lead to apposition, adhesion, and then invasion inside the endometrium. Okay? That's we'll come and discuss in detail. And then starts the trophoblastic differentiation to cytotrophoblast and syncytiotropoblast, or inner cell mass or outer cell mass. Inner cell mass will produce the embryo, outer cell mass will produce the placenta that will keep on supplying the blood. Is it clear till now? Anybody, any doubt? No. So we will get into the next. So clinical correlation will come later on. Gametogenesis, the male gamete, female gamete. You must have learned in your physiology, right? You must have learned it. So we will not go in very detail because we have a long lecture today. So these are the learning objectives, these are the headings we will be discussing about. We will understand the cellular and molecular events in gametogenesis. We will describe the spermatogenesis and oogenesis, and we will see the hormonal regulation pathways. And we will discuss the folliculogenesis and seminiferous tubular microanatomy of male and identify some clinical disorders related to gametogenesis. So, to talk about it is the process. Gametogenesis is a process by which diploid germ cells form the haploid gametes. So, 46XY has become 23X or 23Y, which is waiting to meet 23X from the female. So that is called haploid gametes. And it occurs where? It occurs in the male gonads. Male gonads are the testes, and in case of female, it occurs in the ovaries. So the 23 eggs is produced in female gonads, that is the ovaries, and 23 X oblique 23Y, that is formed in male gonads. Okay? That includes some mitosis, meiosis and cytodifferentiation. As I spoke, mitosis, there is a dividation process, we will come to that. That whole process of making this 23X or 23Y, that includes mitosis as well as meiosis and the cellular differentiation or cytodifferentiation. By what? By the endocrine as well as paracrine and autocrine factors. Okay? There are different factors, different hormones acting in different levels to make this male and female haploid cells or gametes. Okay? So, what is primordial germ cells? The primordial germ cells are the cells, they are derived from epiblast and they migrate to genital ridge during embryonic period in female at around 4 to 6 weeks of development. Okay? These are the certain cell lines which are derived from epiblast and they migrate to genital ridge during embryonic period by around 4 to 6 weeks of embryonic period. They reaches there and they waits there, and they are mitotically active. They can divide. Mitotically active means those cells, primordial germ cells in a female embryo, they reaches the genital ridge coming from epiblast and they are waiting there like this. But they have a capacity for dividing themselves. That's called mitotically active. Any defects in migration of this cell will give rise to germ cell tumor in female. Okay? Now coming to the spermatogenesis. We'll go little first on this. It occurs in the seminiferous tubules and the duration is approximately 64 days, and it contains the different phases like spermatocytogenesis, meiosis and spermiogenesis. Did you learn it in your physiology? You must have read it. It's just a revision. So I'll go a little fast. So coming to the seminiferous tubular microanatomy, it has got germinal epithelium that contains Sertoli cells as well as Leydig cells. Okay? What does these cells do? The Sertoli cells, they support the blood and testis barrier. Like we have a blood-brain barrier, we have a blood-testis barrier. Okay? So everything from the blood cannot reach the testis. Everything from the blood cannot reach our brain. So we have a filtration system. That filtration system, here we call it a blood-brain barrier. There we call it a blood-testis barrier. And it produces anti-Mullerian hormone as well as inhibin. Okay? And the Leydig cells, the main function of these Leydig cells are the testosterone secretion, the male hormone. Okay? Now the spermatogonia, they are A, pale and B types. When there is a mitotic division, this is the small pathway we are showing. The mitotic division will first give rise to primary spermatocytes and the stem cell renewal will take place that will maintain the fertility later on. Coming to the meiosis, the primary spermatocytes, you learn it properly, you have learned it in physiology. This is the spermatogonium which breaks down to form the primary spermatocyte. So the primary spermatocyte, how much is the chromosomal pattern? This is 46XY. When it further breaks down to secondary spermatocytes, okay, this is the first meiotic division here. Okay, after this primary spermatocyte containing with 46XY gets the first meiotic division. Meiosis means half. They will give rise to this secondary spermatocyte. What is the secondary spermatocyte? That contains either 23X or 23Y. Okay? And then that further meiosis 2 will give rise to these spermatids. Okay? So here the meiosis 1, so this 46XY has become 46X and 46Y, and then further this got divided into spermatids with 23X or 23Y. Is it clear? it was 46XY, okay, spermatogonia. Then first meiosis, after the first meiosis becoming half, 23X or 23Y. Now that 23X or 23Y coming here, it has divided into further. That's called meiosis 2, okay, and finally giving rise to spermatozoas. So the second meiosis will give rise to spermatids, whereas first meiosis will give rise to secondary spermatocytes. Is it clear till now? Okay. And there will be no cytoplasmic division at this moment. Cytoplasm will remain as it is, which will be later on shared with the oocyte. Now, coming to spermiogenesis, the transformation of spermatids to spermatozoa is known as spermiogenesis. Okay? And then starts the formation of acrosome, that cover of the sperm head, and the flagella. It has to move like this. Okay? So you have seen the fish, they are moving their tail like this and they are going further. So sperms are also like this. They have got a tail, they move it like this and they swims up. Okay? That's the flagellar action and nuclear condensation. So during this time the nuclear condensation starts. Okay? And they start shedding of excess cytoplasm. In the previous slide, there was no change in cytoplasmic amount, the amount of cytoplasm which no change. Now when it is coming to spermiogenesis, it's a developing of spermatozoa from those spermatids. In the previous slide we read about up to the development of spermatids. Okay, now coming to the spermatozoa, that's the mature sperm. And when the spermatids become the spermatozoa, there is shredding of the cytoplasm. Excess cytoplasm is shed out. Okay? What is spermiation and epididymal maturation? Well, after this maturation, it is released inside the lumen and it acquires the motility in epididymis, inside the testis. And it depends on level of testosterone and androgen binding protein. So the hormonal regulation is almost same. Like in physiology of menstruation, I said hypothalamus, GnRH. GnRH hits the anterior pituitary to release FHS LH. That FHS was working inside the ovary, giving rise to follicular maturation. And once the follicle was matured, then LH surge came and then released the egg. So ovulation took place. Here also almost the same thing, only the gonads are different. Female had the ovaries as the gonads, male has the testes as the gonads. But the mechanism up there is same. Hypothalamus and then pituitary. Pituitary released what? That LH hormone. After the pulsatile release of GnRH from the male hypothalamus, it hits the pituitary, from where it releases the LH that acts on the Leydig cell producing testosterone, and the FHS cell that acts on the Sertoli cell that produces the androgen binding protein and inhibin. Okay? So coming to the oogenesis, is it clear till spermatogenesis? It's very simple actually. If you just understand it without sleeping, then it is very simple. You don't have to read. You can write a big essay on this. Okay? So coming to oogenesis, it begins in the fetal life and they are arrested in prophase 1 until puberty. I said it is 4 to 6 weeks of embryonic period. They reaches the genital reach, the primordial germ cells, and they stays there. And they have a mitotic capacity, but they don't undergo mitosis. Okay? Now, the child is born. Still they are silent. When they start acting, when they reaches the puberty. There is this called cell cycle arrest. Up to prophase 1 and they will not further divide those cells. Okay? And they will start dividing when? When the girl reaches the puberty, those cells now they becomes active and they starts dividing. And the ovarian reserve, we have generally 1 to 2 million at birth. What is 1 million? If you convert in lakhs, you are correct. 1 million is equal to 10 lakhs. So 2 million means around 20 lakhs. Imagine 20 lakhs of cells. And every cell has a potentiality to develop to Graafian follicle. Is it possible? So, a lady is expected, out of this 20 lakhs of cells, a lady is expected to give maximum 350 to 400 ovulation in her lifetime. Okay? So, and it resumes monthly with ovulatory cycle, which I already spoke. Every ovulation there is a running of around 50 to 70 cells. Only one will become the Graafian follicle, it will release, and rest will become atretic, means they will die. That's how from this 1 to 2 million, a lady is expected to ovulate 350 to 400 times in her lifetime. So it goes into different phases. That oogenesis, that's the multiplication, and then the growth to primary oocyte to Zona Pellucida formation, that outer cover, and next reaches the maturation. It occurs meiosis 1 in puberty and meiosis 2 in during fertilization. Okay? So, coming to ovulation and meiosis, the hormonal, what are those hormones? They play a role. It is the LH surge. That completion, after the LH surge, there is a completion of meiosis 1. Once the meiosis 1 is completed, then the secondary oocytes are formed. Okay? And they are arrested at the metaphase 2 till the fertilization. Ovulation हो गया. ठीक है? कैसे हुआ? LH surge हुआ था. At around day 14 of the 28-day cycle, ovulation has taken place because the LH surge was there. After the LH surge, what happened? The meiosis 1 completed of that particular egg cell. Okay? Of that particular egg cell, the meiosis 1 is completed, and they are called secondary oocytes. Now they are waiting. No more further division. And they will wait for metaphase 2. And metaphase 2 will come when? When it reaches the sperm. It meets the sperm. Then only when fertilization takes place, that the completion of meiosis 2. Now they will divide. What they will divide? Then half this set, half will meet with the half of the male, and then a complete set of 46 XX or 46 XY will be formed, and then they from 1 cell to 2 cell, 2 cell to 4 cell, 4 cell to 8 cell, that is called blastocyst, covered with a Zona pellucida, and then starts moving by the ciliary action of the tube and reaching the uterine cavity, and now the hatching takes place. What is that hatching? That hatching is removal of that outer cover, that is Zona pellucida, and now time to get implanted. Is it clear? Okay. Folliculogenesis, we have already spoke about it. There is a primordial follicle, then becomes primary follicle, and then the secondary follicle to antral follicle to the Graafian follicle. The last stage, the Graafian follicle is the biggest follicle that develops and the oocyte is released. So primordial to primary, primary to secondary, secondary to antral follicle, and then comes the Graafian follicle. From antral follicle, around 50 to 70 antral follicle starts running, and one follicle becomes the Graafian follicle or a mature follicle. Rest become atretic. So, and it is FHS dependent, as we already spoke about it. Now, theca interna, androgen, and then from the androgen, the granulosa cells. The granulosa cells and theca cells, I said, the egg cell has got 2 cell layer, that follicle has got 2 cell layer when it is developing, the granulosa and theca. The granulosa produces estrogen, theca produces progesterone. Okay, so this is what we are talking about. So we have already spoke about it. The hormonal regulation will not go deep into it. GnRH to FHS LH, FHS to the ovaries, folliculogenesis, Once the follicle is developed, LH cells from the anterior pituitary break the egg, break the follicle, release the egg. So that's what is this hormonal regulation. It's a very, very simple mechanism and very interesting also. So let us talk a little bit about the comparison. The spermatogenesis versus oogenesis. The development of sperm and development of the ovum. Spermatogenesis is a continuous process. Every day it is taking place. Whereas oogenesis is a cyclic process. It's once in a month. Okay? And there is a production of sperms millions a day versus a single oocyte per cycle. 1 month, 1 egg cell. Okay? And there is the equal versus unequal cytokinesis. And it is lifelong. However, a female has got a limited supply. Okay? So once the AMH, which I spoke about, the anti-Mullerian hormone, it will gradually go down. In a female I am talking about. Because you just imagine you have got a itna sa, just imagine this much of rice you have in your hostel. Everybody is coming and taking, Ultimately it will become empty. So it's over. That is, everybody coming and taking, is every period coming and taking out. Every period coming and taking out those cells. One is ovulating. So the store is decreasing. Once it is over, so the female reaches the menopause. However, in male, it is an ongoing process. So it is produced there. A female comes with a fixed store from her mother once the baby is born. Now, once she reaches the puberty, starts having the period, it starts breaking down from there. So once it is breaking down every cycle from there, that store is gradually going down. Once the completely down, the lady reaches the menopause. So that's why it is called limited supply. Whereas in male, it is an ongoing process. So it's not limited. Let us come to the implantation. So far what we have learned is how the spermatozoas, how the oocytes were formed, okay, and how it got fertilized and transported to the uterus, endometrium. Okay, now let us talk about how it will get implanted there. Is it clear? So my classes started from menstruation, physiology of menstruation, and then ovulation. After ovulation, it was picked up, okay, and fertilization took place. Before fertilization, the formation of spermatozoa and oogenesis we spoke about. Now coming to the next stage of fertilization. Once the fertilization physiology also we have learned, now fertilization has taken place. Now this has to come to the uterus and get implanted to develop a embryo or a fetus. So let us talk a little bit about the implantation. The implantation is the process by which the blastocyst, which I spoke little before, the blastocyst attaches to and invades the endometrium. Who makes the endometrium ready? Good. It's the estrogen that produces the bed. इसका मतलब तुम लोगों को समझ में नहीं आ रहा है. इतना इजी करके मैं समझाने का कोशिश कर रहा हूँ. इतना इजी तो मैं भी नहीं समझा था जब मैं इस टेबल में बैठा हुआ था during my MBBS. Okay. So, estrogen prepares the bed for the embryo to come and get implanted. Okay. That bed is called endometrium. Okay. And that corpus luteum, from where the egg was released, that corpus luteum produces, you sit down, produces progesterone for whom? To support that embryo which is formed and keeps on supporting that embryo till the placenta of that embryo is formed, and that placenta is capable to give enough beta hCG to support that pregnancy. And then gradually the support from the corpus luteum is withdrawn by around 12 to 13 weeks. That's 3 months. By 3 months the placenta is completely formed. It can take care of its own baby, own fetus. I don't need support progesterone from the corpus luteum. So corpus luteum goes down. Okay. So implantation is the process by which the blastocyst attaches and invades the endometrium, that inside layer of the uterus. Is it clear to all of you? So, the endometrium, which occurs around 6 to 7 days after fertilization. Roughly it is 4 to 9 days. Okay? The fertilization, where does the fertilization take place? Ampullary part. And once the fertilization took place in the ampullary part, now it takes approximately 9 days to reach the endometrium. How it reaches the endometrium? Bolo. How it reaches the endometrium? Ciliary motion and the peristaltic movement, both together, and it reaches the endometrium, which requires a synchronized embryonal development and hormonally primed endometrium. What is that hormonally primed endometrium? That is estrogen primed endometrium. Okay? Now comes the different phases of getting attached with the endometrium. What are those phases? We call those phases as apposition, adhesion and invasion. Okay? Now once the blastocyst is formed by day 5 after fertilization, the development of inner cell mass, which I spoke little before, there is an inner cell mass that produces the embryo, and the outer cell mass or the trophoblast that produces the placenta. Starts dividing. Abhi tak kya tha? One cell became two cell. Before one cell, there was a sperm cell and there was an egg cell. They met together, broke the Zona Pellucida, ZP3, ZP2 enzyme, protein, ZP1, ZP3 and ZP2 and protein entered that, sharing was there. Now the embryo was formed, single cell, it become mitosis 2. Started mitotic division, 1 cell to 2 cell, 2 cell to 8 cell, 2 cell to 4 cell, 4 cell to 8 cell, and that 8 cell is called blastocyst. Now the blastocyst traveled from ampullary portion to the uterus with a hard cover around it. That's called Zona Pellucida. And now the hatching took place, where once it reached the endometrium. What is that hatching? Hatching means removal of that Zona Pellucida cover, and now it is ready to get implanted inside. Now it is getting implanted inside and started developing two kind of cells. One is inner cell that is gradually start producing the embryo, and the outer cell that will start producing the placenta. Is it clear? Now the Zona Pellucida thinning and hatching that allow the trophoblastic contact with the endometrium. Trophoblastic cells are the embryonal cells. They need to come in contact with the female endometrium, mother's endometrium. Okay? And then only the development of the communication will develop, the vessel communication. Okay? Now that Zona Pellucida or the outer cover is released, that's called hatching, and now the trophoblastic cells are in contact, directly in contact with the endometrium, the inner layer of the uterus. Okay? And now start the differentiation of trophoblastic cells and syncytial trophoblastic cells. That produces that syncytial trophoblast will produce the beta hCG hormone that supports the pregnancy, prevents it from falling down from the implanted site. Now, endometrial preparation for implantation, which I already spoke about it, the progesterone converts the endometrium to secretory endometrium. Initially, who was the main game player before ovulation took place? Who was making the endometrium very thick and vascular, very softy, velvety? Estrogen. Now ovulation has taken place. Graafian follicle has ruptured. Corpus luteum has already formed around day 14-15. Now the progesterone starts secreting from that corpus luteum. That progesterone makes this endometrium more secretory in nature, making it more what is that called? Secretory means secretory. I don't know the Bengali or this thing. More secretory in nature, which will increase the glandular secretion. What are those glands? Those are endometrial glands. those endometrial glandular secretion will start increasing. It will become more cozy, more slippery environment for the baby. And there will be stromal edema. And pinnapods formation starts. This was the blastocyst. Hatching हो गया. अभी endometrium. Then endometrium becomes more secretory because of progesterone coming from the ovary. What in the ovary? That is the corpus luteum in the ovary. That progesterone is making that endometrium more secretory. What those secretory things are the endometrial glands, and they starts making the pinopods. Okay? That pinopods marks the window of implantation. Whenever there is a formation of pinopods, that means one is ready to receive the other. Okay? That's called the window of implantation. Okay? That is around day 20 to 24. Okay? And which enhances the vascularity and the development of spiral arteries. Spiral means teramera. Okay? Those, why they are teramera, we will come to that later on. So, the vascularity is increased. Otherwise, the blastocyst will eat what? It will need only blood. And in the blood, what it will get? It will get nutrients, it will get oxygen. Okay? So, it needs to develop the vascularity with the mother, vascular communication with the mother, so that, and there is a development of spiral arteries from the maternal side, which will start communicating with the blastocyst or the embryo. So, what are the molecular level that actually plays role in all this physiological process? There is an integrin, there is a protein that supports the adhesion between the blastocyst and endometrium. It integrates, the integrin protein integrates. What is integrates? It integrates the blastocyst into the endometrium. Okay? And there is a leukemia inhibitory factor which is essential for successful implantation. If it is not there, there will be a rejection. Okay? The embryo will come, the blastocyst will come, but it will not get attached with the endometrium. Okay? There are cytokines and growth factors that guide the trophoblastic invasion. What are those trophoblasts? Those are the fetal cells. Syncytiotrophoblasts and cytotrophoblasts. They have to invade the maternal vessels. Okay? If they are not embedded in the maternal vessels, they will not get the blood from the mother, and if the blastocyst doesn't get the blood from the mother, it will not survive. It will get aborted by itself. Okay? So the cytokines and the growth factors which guide the trophoblastic invasion, it helps those trophoblasts ki aaja bhai andar aaja, andar aaja, andar aaja. Under where? Maternal vessels. So once they enter the maternal vessels, the blood from the mother starts flowing toward this blastocyst. And it keeps on growing. The embryo starts growing. Okay? There is a metalloproteinases which enable the stromal penetration. It helps in further penetration of this stroma. Okay? The different stages are apposition, which is a loose initial interaction between blastocyst and endometrium. तुम्हारा guest आया, first उसको gate में मिला. Hello, how are you? So, this is hello, how are you? The blastocyst has come, the endometrium is waiting and telling hello, blastocyst. So, that's called apposition. It is the first interaction between the embryo and the endometrium. Okay? Estrogen primed endometrium, which was made secretory by progesterone coming from corpus luteum. Now the embryo has reached here. Now the endometrium is telling, hello, bhai, how are you? I am waiting for you. So that's the first interaction between the blastocyst and the endometrium, which further leads to adhesion. As I said, integrin. So, integrin mediated form attachment. बस लगा दिया उसके साथ, किसके साथ? एंडोमेट्रियम के साथ. किसको लगाया? ब्लास्टोसिस्ट को लगाया. Okay. Now comes the invasion. What invasion? That maternal vessels are like this. Now the syncytiotrophoblastic cells erode the epithelium and the stroma starts entering, invading the endometrium and the maternal, gradually the maternal vessels to establish a communication between the mother and the embryo. Thus lead to formation of lacunae and there is a start of early maternal blood flow. Is it clear? Now there comes the trophoblastic differentiation and invasion. What is those trophoblasts? Cytotrophoblast and syncytiotrophoblasts. Okay. Now comes the cytotropoblastic proliferation and that forming the anchoring villi. Villi means something called projections. Anchoring means holding. Okay. You know the ये शीप को एंकर करते हैं ना, उसको वहाँ पे रुक के रखते हैं. So these villi, we have got intestinal villi also. So villi are like also ciliated projection kind of thing that is waiting there to anchor that. I will not leave the embryo. I will keep it holding with the endometrium. Okay. So cytotrophoblasts proliferate. What are those cytotrophoblasts? Cyto and syncytio both. Proliferate and form anchoring villi to anchor the embryo, to hold the embryo. Okay? That syncytiotrophoblasts produces the HCG to maintain the corpus luteum. As I said, the syncytiotrophoblasts starts producing the HCG hormone that supports, in one way it supports the embryo, on the other side it keeps alive that corpus luteum which was actually producing the progesterone to support the initially the priming of the endometrium and then once the implantation took place, now supporting the pregnancy. Okay. So the beta HCG which is coming from syncytiotrophoblasts does two things. It prevents the pregnancy loss and also support that corpus luteum so that by keep on giving me progesterone. Is it clear? That decidual reaction transforms the stomal cells into secretory decidual cells. Decidual cells are the basal cells in the endometrium. They become more secretory. And there is a controlled invasion that prevents the deep penetration into the myometrium. We are talking about endometrium. We are not talking about myometrium. Myometrium is the muscle layer of the uterus. Okay? Endometrium is the inside layer of the uterus. Endometrium, myometrium and the serosal layer. So this controlled invasion prevents those invasion to the muscle layer. Because if it reaches the muscle layer, then comes the placenta accreta percreta. Okay? So those will teach you later on. So there is a controlled invasion so that invasion remains within the endometrium and does not reaches the myometrium. If it reaches the myometrium, it will be very difficult to separate the placenta from the myometrium and there will be a huge amount of bleeding after delivery. Okay? That early placental formation, so it has come and it got attached with that anchoring villi with the support of all the hormonal system. Now it is strongly attached with the mother and it has developed the vascular communication with the mother. Now comes the placental formation. Now it needs to develop its placenta from the outer cell mass. Okay? So how does it develop? There is a primary villi formation that becomes gradually develops into secondary villi. Okay? And then it becomes the tertiary villi. And the tertiary villi ultimately gets into vessels, develop into vessels. Villi are the projections. So primary to secondary, secondary to tertiary. Once it is tertiary, it starts communicating with the maternal vessels, myometrial vessels. So chorion frontosome becomes the fetal placenta and the decidual vessel becomes the maternal placenta. Okay? And it ultimately develops to establishment of feto-maternal circulation. Okay? Anything else? It's very simple. We started from the ovulation, menstruation, from the hypothalamus, then pituitary, then ovaries. In ovaries, day 14, egg release. Then that egg got fertilized where the ampullary part of the tube. Then it gradually is transported to uterus and it got implanted inside the uterus. Now it has developed in the placenta. Now the baby will grow. Okay? Finally we will wait for delivery. Is it clear? Anybody any question? कुछ समझ में आया? Anything you understood? If you have understood, you will have some queries. Or if you have understood everything, there will be no queries. Which one should I take? You understood everything. So please go and read just once. Okay? That will actually, you will have an idea how does it work. It's a very, very beautiful physiology. Okay? Chalo.